हेलो दोस्तों, वेलकम टू बायो बिट्स। मैं डॉक्टर सुनील पाटिल आपका बायो बिट्स में स्वागत करता हूं। इससे पहले लास्ट वीडियो में हमने बायोमॉलिक्यूल्स पार्ट फर्स्ट देखा था, जिसमें हमने कार्बोहाइड्रेट इन डिटेल स्टडी किया था। इस वीडियो में हम प्रोटीन एंड अमीनो एसिड इन डिटेल स्टडी करेंगे, जिसमें अमीनो एसिड स्ट्रक्चर, अमीनो एसिड के ज्विटराइन फॉर्मेशन और क्लासिफिकेशन ऑफ अमीनो एसिड according to essential, non-essential, polarity, according to the metabolic properties of the amino acid and different ways. Then we are going to see how the peptide bonds are formed. इस लेक्चर में हम प्रोटीन्स का क्लासिफिकेशन भी देखने वाले हैं। अगर ये लेक्चर आपको अच्छा लगे तो इसे सब्सक्राइब करना ना भूलिए, इसके साथ ही शेयर कीजिए और लाइक कीजिए। तो चलो चलते हैं, हम पहले प्रोटीन वर्ड से शुरुआत करते हैं। द प्रोटीन, the term protein coined by the Berzelius in 1983. Another scientist Mulder referred the term protein to the complex organic nitrogenous substances found in the animal and plant cells. Proteins are polypeptide. They are linear chains of amino acid linked by peptide bonds. Then we are continuing the properties of protein. Proteins are large molecules containing amino acid units ranging from 100 to 3000. Protein have high molecular weight. In protein, amino acids are linked together by peptide bond, which are join the carboxylic group of one amino acid with the amino group of another. A protein molecule consists of one or more polypeptide chains. Protein can contain any one or all 20 naturally occurring amino acids. Then we are going to see in detail the amino acids. The amino acids are building block of proteins. Amino acids are organic compounds containing an amino group NH2 and an acidic group COOH or carboxylic group as the substituent on the same carbon, that is called as the alpha carbon. Hence the amino acids are also called as the alpha amino acid. There are 4 substituent groups occupying the 4 valence positions of the carbon. These are hydrogen, carboxylic group, amino group, and fourth one is called as the variable group, as it is different for different amino acid, designated as the R group. Based on the nature of the R group, there are many amino acids, more than 300 are known. However, these those which occur in the protein are only 20 amino acid, or only 20 types of amino acid occur in the proteins. We are continuing the characteristic of the amino acid. The amino acids in our bodies are referred as the alpha amino acid. Why they are referred as the alpha amino acid? The reason is that the central carbon is in an alpha position in relation to the carbonyl carbon. The carbonyl carbon from the carboxylic group, it is carbonyl carbon, and the adjacent to this, it is called the alpha carbon. Adjacent to the alpha carbon is called the beta carbon. Adjacent to it is called the gamma carbon, then delta carbon, then epsilon, and so on. The carbon adjacent to this carbonyl carbon is alpha carbon. Each carbon is designated with the different letters of Greek alphabet. So due to this reason, it is near to the or adjacent to the carbonyl carbon, it is called the alpha carbon, and amino acids are alpha amino acid due to this reason. The structure of amino acid, this is alpha carbon, which having the 4 valence groups. The one is occupied by the amino group, that is NH3, then one occupied by the carboxylic group, then third one occupied by the hydrogen, and fourth one it is different. It is called as the variable group, or it is called as the R group. If this R group it is hydrogen, then the amino acid is called as the glycine. If it is CH3, it is called as the alanine. In this way, amino acid are classified, or types of amino acid, it is depend on the variable group, that is the R group. Then we are going to study the amphoteric properties of the amino acid. At physiological pH, that is 7.4, carboxylic group is dissociated, forming negatively charged carboxylate ion, that is COO, and amino group is protonated, means accept the electron or hydrogen, protonated, forming positively charged NH3. This condition forms the Zwitter ion. This is the carboxylic group, this is the amino group. The carboxylic group loses electron or hydrogen, and it is protonated to the amino group. So it become positively charged NH3 and it become negatively charged carbonylate ion. So in this way the Zwitter ions are formed. Amino acid shows buffering capacity. Alpha amino acid are having the carboxylic acidic and the basic amino group. These two functional groups allow them to act as a buffer. So this is one characteristic. Amino acids are classified into two large categories or grouped into two large parts, that is non-essential and essential. First of all, we are going to see the non-essential amino acids. These are the amino acids that our body can create out of the chemical found in our body. So body can create or prepare the amino acid from the chemical which are present in our body. They are called the non-essential amino acid. Here are the different amino acids. These are the name of the non-essential amino acid. They are synthesized from the chemical present in our body. Alanine from the pyruvic acid, arginine, then aspargine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. Then second part is the essential amino acid. The essential amino acid cannot be created in our body. Therefore, only way to get them, it is the food. So must be consumed. They are called the essential amino acids. They cannot prepare in our body or not created in our body, so they must be consumed with the food. The essential amino acids are the histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. These 9 are the essential amino acid and 11 are the non-essential amino acid. So amino acids are essential or non-essential. Then we are going to see some test of amino acid and proteins. Ninhydrin test, it is positive test for amino acid, not a peptides. It is particularly for amino acid. All amino acids gives the blue color. There is one exception, that is proline, which gives yellow color. The second test is the xanthoprotic test. Amino or hydroxylic group gives the color. It is the example is the tryptophan and tyrosine. Biuret test, it is positive test mainly for the peptide bonds. The Sanger reagent, it is used to determine the end terminus of the amino acid polypeptide or peptide. So these are few tests for the proteins and amino acids. Then we are going to see the classification of amino acid. The amino acid can be classified into acidic, basic, and neutral. So acidic amino acids at physiological pH will carry negative charge and 2 or more carboxylate ion. Carboxylate ion groups are present in its structure. They are called the acidic amino acid. The example is the aspartic acid and glutamic acid. So you can see the carboxylate structure, this one and this one. 2 carboxylate structures are there. It is one characteristic feature of the acidic amino acid, and they are negatively charged, and this is a single positive. So amino acid become negatively charged. So they are called the acidic amino acid. In the same way, the basic amino acid contain 2 or more NH2 groups. So they are positively charged. Nitrogen atoms that acts as a base and can bind to the proton. At physiological pH, the basic amino acid will be positively charged. Example, lysine, arginine, histidine. You can see in the lysine, this is amino group, this is another amino group. Then in the arginine, you can see the amino groups. In the histidine, this is amino group and this is NH, this is another NH. So it is the basic amino acid. Next is the neutral amino acids. Neutral or uncharged amino acid that are overall neutral molecules but contain charged ends called as Zwitter ions. Neutral amino acid where R groups are glycine, for hydrogen is the R group, alanine, CH3, valine, leucine, isoleucine, they are isopropyl groups, and in leucine they are isobutyl. Then cysteine and methionine, they are sulfur containing amino acid. Then serine and threonine, they are hydroxy amino acids. Then phenylalanine, tyrosine, and tryptophan, they are aromatic amino acid. Phenylalanine, tyrosine, and tryptophan, they are aromatic amino acid. Tryptophan contain the amino acid. Proline consists the amino acid. Proline amino group enters in the ring formation in the proline. So instead of amino acid, proline is called as the amino acid. So these are the neutral amino acids. So classification according to polarity or side chain. Polar amino acids in which R group contain polar hydrophilic group, so can form hydrogen bond with the water, where R group with OH group, SH group, amide group, NH2 group, these are the polar amino acid. These are examples of polar amino acids: serine, threonine, cysteine, tyrosine, asparagine and glutamine. These are the polar amino acids. Then non-polar amino acid, R group is alkyl hydrophobic group. It is the difference, which cannot enter in hydrogen bond with the water. Or there are 9 amino acids which are non-polar. These are the 9 amino acids which are non-polar. The glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline and methionine. Then protein can be classified on the metabolic way. They take part in the metabolism. Metabolic classification, or it is degradation product of amino acid. According to them, they are classified into 3 categories. One is the ketogenic amino acid, which gives the ketone bodies on the degeneration or degradation. The lysine and leucine are the ketogenic amino acid. They are only pure ketogenic amino acid. Second one, the mixed ketogenic and glucogenic amino acid, which give both ketone bodies as well as the glucose. Examples are the isoleucine, phenylalanine, tyrosine and tryptophan. And third one is the glucogenic amino acid, which gives only glucose on the degradation. They include the rest of amino acid. These amino acids are catabolized on catabolism. They yield products that enter in glycogen or formation or glucose formation. So these 3 are the types according to the metabolic classification of protein. Then we are starting the main part, that is peptide bond formation. The peptide bond, or it is also called the amide bond, it is a covalent chemical bond. It is formed between 2 molecules when the carboxyl group of one molecule or one amino acid reacts with the amino group of other amino acid, causing the release of a water molecule. H2O is released. Hence the process is also called the dehydration synthesis, or it is also called the condensation reaction, as water molecule is released. As usually occur between amino acid, the result is CONH bond. It is called the peptide bond, as resulting molecule is the amide. The 4 atom functional group is the CONH, is called the peptide link. We are going to see the peptide link in detail. This is one amino acid, this is another amino acid. This is hydroxyl group of COH and this hydrogen. Remaining is the C double bond O and NH, it is remaining. They get joined and this water molecule is released. So this peptide bond C double O and NH, the peptide bond is formed, that is the peptide bond formation and release of water molecule. The free end, it is with the amino group, it is called the N terminus, and where the carboxylic group is free at the right side, it is called the C terminus, or it is called the amino terminus or N terminus. It is called the carboxylic terminus or C terminus. So N and C terminus are present. N terminus it is always towards the left side, and C terminus it is shown at the right side of the polypeptide. Each polypeptide chain starts on the left side by a free amino acid of the first amino group called the N terminus. Each polypeptide chain ends with the right side by their carboxylic group of the last amino acid called as the C terminus, which I have already told you. How peptide bonds are formed. Suppose this is one amino acid, we are only showing here the amino group and carboxylic group, not the hydrogen and R group. So from carboxylic group of the first amino acid, that is hydroxyl OH, and from this amino group one hydrogen, it forms the water molecule, that is called as the dehydration, and it forms the peptide bond. C double bond O NH is formed. This bond is called the peptide bond. In this way peptide bonds are formed and amino acids are from the polypeptides. Then we are going to see the structure of protein. Functional protein is not just a polypeptide chain, but one or more polypeptide precisely twisted, folded or coiled into molecule of unique shape or conformation. This conformation is essential for some protein function. Example, enable a protein to recognize or bind specifically to the another molecule, hormone and receptor, enzyme and substrate identification, then antibody and antigen reactions. According to this conformation, there are 4 levels of protein structures, that is primary, secondary, tertiary and quaternary structure. We are going to see one by one. First we are going to see the primary structure of protein. The linear sequence of amino acid in polypeptide chain of protein form its primary structure. The primary structure is created through the linking of amino acid by peptide bond. The linking is accomplished by formation of peptide bond. So this is the continuous linear linking of the amino acid by peptide bond. This is called the N-terminus residue. In the polypeptide chain, the amino acids are referred as the residues. So this is the amino group of the first residue, that is, so it is called the N-terminus, or it is left hand side, and in the right hand side or end one, where is the carboxylate ion is it is exposed, group is exposed, it is C-terminus. So this is the structure of primary structure of protein. Each peptide chain will have an amine end and a carboxylic end. Each amino acid residue at the left side of the polypeptide is named as the N-terminus residue, and at the right side it is termed as the C-terminus residue. Then we are going to see the secondary structure. Due to hydrogen bond formation between the hydrogen of NH group of peptide bond and carbonylate oxygen of another peptide bond. This bond formation occurs between the peptide bonds of the polypeptide. So hydrogen bond is formed between the hydrogen of NH group of one peptide bond and carbonyl oxygen of another peptide bond. So this is called as the secondary structure. According to H bonding, there are 2 main forms of secondary structure. One is called the alpha helix, another one is called the beta sheets. Alpha helix, it is the helical structure resulting from hydrogen bonding between one peptide or first peptide bond and the fourth one, means in between 3 amino acids are clearly present. So in this way peptides are formed, so it form a helical structure. Due to this, after the 4th amino acid, at every 4th amino acid there is a hydrogen bonding, it forms the alpha helical structure. And second one is the beta pleated sheet, is another form of secondary structure in which 2 or more polypeptide, different polypeptide chains, it happens in the same polypeptide, it happens in the 2 or more polypeptide, they are linked together by hydrogen bond of hydrogen of NH, same thing, hydrogen bond of one chain, it is different thing, and carbonyl oxygen of adjacent or another chain. So in this pleated structure is formed between the different polypeptide. These are the 3 different polypeptide. In this way, in the hydrogen of one peptide bond from the NH and the oxygen of the another peptide bond, that is COO, there is hydrogen bond formation. In this way, between different polypeptide, the hydrogen bonds are formed and pleated sheet is formed. It is called the beta pleated sheet or beta sheet. So we are going to see again the secondary structure in detail, that is this is the alpha helix. After every 3 molecule there is hydrogen bond is formed. It forms in this way helical structure, it is called as the alpha helix. And in this way we have already seen the structure alternate, the peptide bond, in between peptide bond hydrogen bonds are formed between the different polypeptide and beta pleated sheet is formed. So alpha helix and beta sheet, these are the 2 forms of the secondary structure of the protein. Then we are moving on the tertiary structure of the protein. The tertiary structure, it includes, it determined by the various variety of bonds among R groups and between R groups. In secondary structure they are between the peptide bond, hydrogen bonding. In tertiary structure the bond formed between the R groups, and of same polypeptide or other polypeptide backbone. These different bonds are formed, which are hydrogen bond among polar side chains. The weak interactions include the ionic bond, between charged R groups, hydrophobic interactions among the hydrophobic R groups, strong covalent bond or including the disulfide bonds or bridges between the cysteine residue, between the sulfohydryl groups, SH group of cysteine monomers, stabilizing the structure. You can see in the figure, this is disulfide bridge between the cysteine residue, then salt bridges are formed, then hydrophobic interaction or weak interaction are formed. In this way, different amino acid ionic bonds are formed, hydrogen bonds are formed, and tertiary structure includes these bonds. We are also going to see the detailed structure of tertiary structure. Overall tertiary structure showing the, you can see this alpha helix structure. Then pink color, it is alpha helix. Yellow color, it is beta sheets. Yellow color, it is beta pleated sheets. White color, they are primary proteins. So primary and both secondary togetherly form the tertiary structure. These tertiary structure include this. These are same bonds we have already seen. That is, the hydrogen bond is formed. Then disulfide bridges, ionic bonds are formed. Then weak interaction or hydrophobic interactions are formed, or van der Waals processes are there. So this is the tertiary structure. Then we are moving on the quaternary structure of the protein. Quaternary structure of protein, it forms by the aggregation of two or more polypeptide subunits held together by non-covalent interaction like hydrogen bond, ionic or hydrophobic interactions. So the example of protein having quaternary structure. So two or more polypeptide subunit held together by non-covalent interactions like hydrogen, ionic or hydrophobic. So examples, they are also classified into quaternary. They are again dimeric, trimeric and tetrameric. The dimeric example, dimeric means 2 polypeptide, trimeric means 3 polypeptide, tetrameric 4 polypeptide together, or they form bonds together. Example of dimeric quaternary structure is insulin, 2 polypeptide alpha and beta chains. It is called the dimeric structure. Trimeric collagen is the fibrous protein. The 3 polypeptide chains supercoiled. This is structure of collagen fiber. Then tetrameric, a common example is the hemoglobin. It is a globular protein with the 4 polypeptide chains together. So this is the quaternary structure in detail. So primary, this is from the secondary structure. Primary formed by hydrogen bonding, secondary. Then secondary having 2 types, alpha helix and beta sheets. Then these 3 togetherly form the tertiary structure. Different polypeptides form the quaternary structures. So then we are moving to the classification of protein. Protein can be classified into 3 categories, that is simple protein, then conjugate protein and derived protein. The first one is the simple protein. The proteins on hydrolysis gives only amino acid. They are called the simple protein. On hydrolysis they give only amino acid. They are simple protein. Examples, first one is the albumin and globulin present in egg, milk and blood. They are protein of high biological value, that is, they contain all essential amino acid and they are easily digested. Then globins, they are basic protein rich in the histidine amino acid. They are present in hemoglobin of RBC and combined with the DNA. Then scleroprotein, they are structural protein, keratin, collagen and elastin. They are the simple proteins. Then gliadins, protein present in the cereals. Then we are going to see the conjugate proteins. The protein on hydrolysis gives protein part as well as non-protein part. They are called as the conjugate protein. We are going to see some examples. Lipoprotein, these are the protein of conjugate with lipids. They are so called as the lipoprotein. The functions, enter in the cell membrane structure and helping lipid soluble substances to pass through cell membrane, help lipid to transport in blood. Phosphoprotein, protein conjugate with the phosphate group. Lipoprotein, protein plus lipid. Phosphoprotein, protein plus the phosphate group. So phosphorus is attached to the hydroxyl group of serine or threonine, example casein of milk, or vitaline of the yolk are the examples of phosphoprotein. Then glycoprotein, proteins plus sugars, so they are called the glycoprotein. Example is the mucin, some hormones such as the erythropoietin hormone in cell membrane structure, in blood groups the glycoproteins are found. Then nucleoprotein, these are the basic protein histones, they conjugate with the nucleic acid, they are called the nucleoprotein. If the histones conjugate with the DNA, they are called the chromosome. If histones conjugate with the RNA, they are called the ribosomes. So chromosome, DNA plus the histones, ribosome, RNA plus the histone. So they are called the nucleoprotein. Then metalloprotein, these are the protein conjugate with the metal ions like Hb, iron, copper, then zinc, like in insulin. The chromoprotein, these are the protein conjugate with the color or pigments. So melanoprotein in hair and iris, these are the conjugate protein. Then we are moving on the last part, that is the derived protein, which produced from the hydrolysis of simple protein. They form by the hydrolysis of simple protein. Example is the peptone, from hydrolysis of albumin, and gelatin is they form from the hydrolysis of collagen. So in this way 3 types are there: the simple protein, conjugate protein, and derived protein. Simple protein on hydrolysis only gives the amino acid. Conjugate protein on hydrolysis gives protein as well as non-protein part. And derived protein, they form from the hydrolysis of simple protein. So they form different structure. They are example is the peptone and gelatin. So this is about the protein and amino acid. If you like this video or this lecture, subscribe it, share it, like it. इसे सब्सक्राइब करना ना भूलिए. So have a nice day. See you again. Thank you very much.